That's what he came up with, but it turned out not to be correct. And incidentally, Linus Pauling, I think he did his work at Stanford University, and he's a little bit more famous for vitamin C. He was the first one who said that vitamin C may help prevent colds. And I think the latest research on that, if I remember, is they say that vitamin C does not stop you from getting a cold, but if you do get a cold, it shortens the amount of time that you have the cold. So he was partially right in that way also. But anyway, the two researchers who actually discovered the double helix structure of DNA were these two right here. This is James Watson. He was an English researcher, and this is Francis Crick. He was an American researcher down there on the right. And they were working, I think, at Cambridge University in England. And again, they did not do any physical experiments with DNA in test tubes or anything. They, just like Linus Pauling, got some molecular models and just started working with them to try to figure out how they can get the DNA Gs and As and Ts and Cs to fit together in ways that were consistent with other people's experimental results. And there's kind of an interesting, I guess, controversy about their discovery. Watson and Crick were friends with another researcher, another professor at Cambridge, and working in that other professor's laboratory was this woman here named Rosalind Franklin. And Rosalind Franklin was also trying to figure out the structure of DNA, but she was doing it with some actual experiments called X-ray crystallography. She got some DNA and would shoot X-rays at them, and based on the reflection pattern, you could essentially get an image of what the structures are. It's a very fuzzy image, but this is what she got right here. And it's essentially looking down the barrel of the helix, as if you had the helix like a corkscrew and you're looking right down the barrel. So that if you know how to interpret these X-ray crystallography pictures, it's very clearly helical in structure. And so her boss took her photograph and showed it to Watson and Crick, since he was friends with them. And they saw that, they said, oh, okay, it's obviously a helix structure, and that gave them their basic idea. And then they started putting their molecular models in a helical shape, and eventually they got the right answer, and they eventually got a Nobel Prize for their discovery. And so, you know, was that underhanded of them to look at her data? Well, I don't know. You know, generally, if you invent something, it's really your boss's property. If you're working for a company, what you do there is the property of the company. And so I guess her boss had the right to share that photograph of her work if he wanted to. But what people have said about Watson and Crick is they should have given her more credit. In the paper that they wrote on the double helix structure of DNA, they did acknowledge her, but in a relatively small way. And a lot of people in retrospect said that they should have given her equal credit, that it should have been Watson, Crick, and Franklin as the official discoverers of DNA. Matter of fact, when I was taking essentially this class back in San Jose State years ago, my professor was really mad at Watson and Crick for doing what they did. And so he would just whisper their names. He would say, DNA was discovered by Rosalind Franklin and Watson and Crick. Kind of cute. But anyway, so in modern accepted biology textbooks, usually she's given equal credit with Watson and Crick. And as I said, they eventually got the Nobel Prize for discovering the structure. She died at a very young age of cancer, maybe from all the X-rays she was working with, I'm not sure. And they don't give Nobel Prizes to people who have died, and so she wouldn't really have been a contender for it anyway. It's kind of a tragic story. In any event, so after their discovery of the double helix structure of DNA, there was a very productive couple of decades where various biologists then figured out how DNA worked, how DNA can reproduce itself inside the cells, and how genes on the DNA are expressed. And so there's the ladder model of DNA. And of course, it's actually twisted in that double helix structure. Anyway, so we've gone over the basics of DNA expression, how the genes get transcribed into messenger RNA, which is what this molecule right in the middle is supposed to be, and then the messenger RNA is translated by the ribosome. I just want to do a real brief recap of that because it's going to become pertinent as we now focus more and more on DNA. And just to give it some context, I thought I would use Mendel's purple flowers as an example. So remember, Gregor Mendel was the guy in the 1860s who discovered the rules of genetics, and so one of the genetic traits he was working with were this trait of purple flower color. And so let's see how a gene can give that physical trait. The gene that gives that purple trait is part of the chromosome. Genes are segments of the DNA in a chromosome. And so let's say this is the gene that confers the purple flower color. When the plant needs to express that gene, to use that genetic information, the two strands come apart like this of the gene, and then this enzyme called RNA polymerase comes along. And remember, the RNA polymerase binds to one of the strands called the template strand, and it rides along it like a train on a train track. And in the process, it makes an RNA strand that's complementary to the template strand. If there's T there, there's an A there. If there's an A in the template strand there, you get a U, because U is like T for RNA. C there gives you a G there, C there, G there. Like you get a complementary RNA strand. And notice then that if you do that, the strand you get has the exact same codons as the non-template strand of the DNA. A TG, a UG, GGA, GGA, CCC, CCC, TGA, UGA. It's really all the same codons, except it's now written in RNA. So you have a temporary disposable copy of one of the strands of the gene, and that's called the messenger RNA. And then that detaches from the gene. It exits the nucleus. Here you see in blue the messenger RNA there. It exits the nucleus and goes out into the cytoplasm, where, remember, it meets up with a ribosome, which is the protein and RNA organelle that's involved in translating the messenger RNA into an actual protein. And there are also these transfer RNAs, these little bits of RNA that shuttle amino acids that are floating around in the cytoplasm to the ribosome organelle. And the tRNAs also have what are called anticodons on their bottom that allow them to match up by base pairing to the codons in the messenger RNA, and that ensures that the correct amino acid for the AAA codon in the messenger RNA gets brought into the protein at the right position at the right time. And so the ribosome takes these incoming amino acids on the transfer RNAs and makes new peptide bonds between them, or forms peptide bonds between them. And that is how the protein is made from the gene. There's the genetic code of all the amino acids and the codons that go with them. And so now a protein is made inside the cell, this guy right here. And proteins, especially enzymes, carry out chemical reactions. And what this particular protein is going to do is manufacture a purple pigment molecule. It's an enzyme that makes purple molecules. So the cell becomes filled with these purple pigment molecules. And so you get a purple cell in the plant. And this is not just happening in one of the plant cells, it's happening in all the cells of the flower. So you end up with a purple flower. So the physical trait, the purpleness in this case, was actually manufactured by a protein, but that protein was made from the instructions in the DNA. And so in the final analysis, we say that it's the gene in the DNA that gives the trait. The protein was just the worker who who happened to be the one that carried out that trait, but the genes give the traits. Well, all right. For the remainder of this chapter, I want to focus in on the chromosomes themselves. Here we go. So if you look at a cell under a microscope, at least in certain times in the cell cycle, the cell's life cycle, you can actually see the chromosomes pretty clearly. They're inside the nucleus.
What do you mean by certain times in the cell cycle?
Yeah, good question. Cells go through reproductive cycles where sometimes they're dividing to make new cells, but other times they're essentially resting and not dividing. And that phases we go through where they divide and then rest, then divide, then rest is called the cell cycle. And at certain time points in that cycle, you can see the chromosomes very clearly as little threads. But at other times in the cycle, they get kind of fuzzy and hard to see is what I'm saying. So when the cell is resting, that's when it expresses the genes, and the chromosomes have to become a little bit more spread out to get off the histone protein. Remember, the histone protein winds them tightly, and so the cell has difficulty expressing the genes when they're wound tightly on the histones. And so when it wants to express them, the DNA comes off the histones, and so you no longer see nice chromosomes. They sort of spread out in the nucleus and get fuzzy.
Go ahead and transcribe.
Yes, exactly. It opens them up so the enzymes can get in there more. That's exactly it. So this is a diagram of a chromosome I was showing you before. The centromere, the spot in the middle where it acts as a handle to move the chromosome around. The part that I'm showing you in red there with the DNA helix inside of it is called the chromatid. The chromatid is the big long piece of double-stranded DNA of the chromosome and the histone proteins that it's wrapped around. In this diagram, I'm not really showing the histone proteins that it's wrapped around, but those are included as part of the chromatid also. So essentially, the chromatid is the same as the chromosome, but not for the centromere. The centromere is not considered part of the chromatid, but it is part of the chromosome. Or I guess you could say it another way, a chromosome is a chromatid plus a centromere together. Okay, now, so here's a cartoon of a cell inside the nucleus. You can see a whole bunch of chromosomes, each with its own little centromere there. As I was mentioning to Rolando just a moment ago, actually, in certain parts of the cell's life cycle, you can see the chromosomes very plainly as little thread-like structures inside the nucleus. But in other parts of the cell cycle, they look like this. They get kind of spread out and diffuse looking, so you can't really see individual thread-like structures of the chromosomes. That's called chromatin, the diffuse, spread out state of the chromosomes. And as I just mentioned, it really comes down to whether the DNA of each chromosome is wrapped on histones or not. These purple little cylinders there represent the histones. So, and the blue band is the DNA. So if the DNA is very tightly wrapped around the histones on the chromosome, you get condensed chromosomes, as they're called, that look like little threads. But if the DNA partially unwinds from the histones like this, which is actually where it spends most of its time, because in this state right here, it's easier to express the genes. There's lots of room there for the RNA polymerase and the other enzymes to get in there and express the genes. In this more spread out state, it's easier for the cell to express the genes. So the cells tend to spend most of their time in this state, but since the chromosomes are partially unwound off the histones, they spread out more in the nucleus, and so you don't see them as individual, tightly wound up, condensed chromosomes. They are chromatin. but you know, all the chromosomes are still there. Their DNA is just not tightly wound up. Here's a nice photograph. This is actually from the root tip of an onion, and you can see all the cells there with their yellow cell wall and the stained nucleus inside them. And notice that in most of the cells, you can't see individual chromosomes. It just looks like sort of a ball of stained DNA inside the cell, like here, and there, and there, and there. But in some of the cells, you can see the chromosomes where they've condensed. So those are the ones where you can see the chromosomes are the cells that are right about to divide. They're the division part of the cell cycle. Alrighty, well, let's see now. Let's zoom in on the nucleus of this particular cell. Take a closer look at the chromosomes. If you looked at pretty much any of the cells in your body and counted the chromosomes, it would come out to 46. And there are a couple exceptions to that. I think I mentioned your reproductive cells do not have 46 chromosomes, and there's a few other cells that it's not this exact number, but don't let those minor exceptions distract you. Pretty much in every cell in your body